So you are at the West Coast? Oh no, Harvard, so East Coast.
I'm on the East Coast now.
All right, so the 6 hour difference in time, I guess.
Yes.
Not too bad.
No, that's actually perfect.
Yeah, just wait a couple of more minutes. Give people some time to come back. So you have about an hour for the whole thing. So I would recommend to do 45 minutes and then maybe have some discussion. I don't know, do you want questions during your talk or do you want it at the end?
I don't mind. So I can take questions during the talk as well as at the end. I'm planning for around 45-minute presentation and then happy to take questions and have a discussion.
Okay, so if somebody posts something in the Q&A, I will alert you to the question.
Yeah, that'd be great.
During the talk, and then you can answer it. If it gets too much, then I'll just let you go until the end, not to interrupt too much. All right, so I think we should be almost ready to start. So I don't see people, but I'm sort of assuming that people are back in the attendee section of the Zoom as well as in the live stream, where we also have had a good number of people watching the talks today. I'm very happy and honored to introduce our keynote for today, which is Dr. Marinka Zitnik. She is a computer scientist studying applied machine learning with a focus on challenges brought forward by data in science, medicine, and health. And she's joined Harvard as an assistant professor in 2019. Before joining Harvard, she was postdoctoral scholar in computer science at Stanford University. And her recent research recently won, for instance, the Bayer Early Excellence in Science Award. And she was named a rising star in electrical engineering and computer science by MIT, and also a next generation in biomedicine by Broad Institute of MIT and Harvard, being the only young scientist who received such recognition in both EECS and biomedicine. So she has already a very distinguished career, and I'm very happy that she joined us today to give a talk called Graph Neural Networks for the Development of Therapeutics. So the stage is yours.
Thank you so much, Max, for this kind introduction and invitation. It's my honor to be here with you today and talk about the use of graph neural networks for the development of safe and effective medicines. So let me begin. Science and medicine crucially depend on the existence of scientific instruments. And while in the past, those scientific instruments were primarily physical devices that have facilitated discoveries, such as microscope. Now in the era of modern data-intensive sciences, we need a new kind of instruments that might not be physical devices anymore, but virtual devices. And myself, as many others in the field, think that those devices will in vital ways depend on knowledge discovery from data. So this is really a fantastic vision to have in the context of data that we see in medicine and health and science, but it's still relatively challenging to realize. So why is it so challenging to realize this vision? I will mention here some of the broad challenges on this slide that we will be then addressing in the rest of the presentation today. So the first challenge is that for most of biomedical phenomena, there really isn't a single data set that one could very simply look at and understand the entire complexity of say, a disease or be able to identify a successful drug treatment. Very often we need to integrate data that is heterogeneous, potentially confounded, that comes from a variety of different resources, including experimental readouts, might be curated, self-reported, and that data spans from molecules all the way to society and behaviors. And so typically does not fit into that large, flat, single matrix data representation. Second point is that in order to be able to really facilitate scientific progress in these other disciplines, we need to be able to generate predictions that are in some way actionable, meaning that they would readily translate to hypotheses that can be validated in the lab and can be validated in downstream experiments in order to maximize the yield of scientific discovery. And so much of the research in my group and my lab is really leveraging graphs and networks as a general language to bring together very heterogeneous data and then learn and reason over those. The way we typically think of that problem is that we are really modeling networks and interconnected data at the level of populations, where we think of various kinds of relationships between individuals, such as patients in a society, down to the level of individual, where within a person there are different kinds of phenotypes or diseases that might codepend, be comorbid, or interact with each other, and all the way down to cellular level, where we have different molecules that interact with each other, that bind to each other. And so we can now build these connections across the scales, and so that allows us to get a unified representation that is multi-scale as well as rich and multimodal, in the sense that it brings together a variety of very different data types and their descriptors. So in today's talk, I will show to you and explain and describe some of the efforts that we have recently made in using networks and graph neural networks in particular to advance and facilitate scientific advance in the area of development of drugs and therapeutics more broadly. So in doing so, I will describe two recent algorithms that we developed. One is called subgraph neural networks. The other is focused on meta-learning for graphs. And then I will mention two important applications in the area of therapeutics. The first is focused on using graph ML for safe and effective drugs. The other is focused on using graph ML to identify treatments for most challenging, never before seen diseases. And finally, hopefully the time will allow me to also mention Therapeutics Data Commons, which is really a resource that we have been developing as part of as a larger consortium that we founded later a few months ago, with the goal of providing machine learning community with a set of ready-to-use data sets, as well as learning tasks that are meaningful, that can facilitate algorithmic innovation in the area of therapeutics. So I hope that will also be useful to many here. I will be mentioning medical images in the sense of using them to make sense of some of predictions that graph neural networks will be made, but much of the talk will then be focused more on graphs and knowledge representations as they're given to us. But I hope that you will enjoy the talk and certainly get something out of it, even though it might be slightly orthogonal to sort of the core imaging area. Okay, first part of the talk, I'll focus on subgraph neural networks as a methodology with the goal of identifying effective drug treatments. Okay, so the motivation for that is that the development of completely new drug and getting a new drug to the market is an extremely laborious, resource intensive and costly endeavor. It takes on average $2 to $3 billion today and over a decade to get a new drug designed from scratch and get it to the market, because because the drug needs to go through a series of validations, iterative development, screening, clinical trials, and then finally regulatory phase in order to be approved so that patients can actually take the drug. And so that is challenging, and faced with the skyrocketing cost for developing new drugs, researchers have been looking at ways to repurpose older ones. What that means is that one would be looking at drugs that are already on the market, or perhaps that have failed early or late stages of clinical trials, and see whether those drugs can be repurposed or repositioned for some other disease. that they were not initially designed for. And so that problem is known as drug repurposing or drug repositioning. And it's a really opportunity that many are pursuing in the hope of expediting, accelerating development of drugs, which has been, for example, especially important in the context of the last year or so because of the pandemic, which I will also mention later in the talk. So the way to think of it, think of this drug repurposing problem at the high level, you can think of it in this way. Imagine we have a very large number of drugs, and we look at all approved drugs across the US or Europe, which might give us tens of thousands of drugs and their various substitutes or variations, which would then be on the order of hundreds of thousands of those. On the other hand, we have the entire range of human diseases as codified by medicine, which would be tens of thousands of diseases. And for some diseases, we have good treatments, and for some other diseases, no good treatments exist yet. The way disease would be then represented, it would be indicated by edges linking drugs to diseases, where an edge indicates a known treatment relationship, meaning the drug is used to treat the disease, or in other words, it is indicated for the disease. So the question of drug repurposing at a very high level, its goal is to then predict what disease a new molecule or a repurposed molecule might treat. So the goal is to identify therapeutic opportunities for that green molecule, in example, and answer whether that might treat the disease on the right. So the key insight to operationalize and this problem and start addressing it is the use of are actual subgraphs. So let me explain why subgraphs are so important for this problem and why they're relevant. So the way we can think of human diseases is that they are not independent of each other, but many human diseases are symptomatically similar. For example, cancers are more similar to each other than neurodegenerative diseases, which are again more similar to each other. So diseases share many of these common molecular underpinnings, which we would formally say they share some common genes that when those genes are mutated, give rise to the disease. On the other hand, also drugs share some commonalities. They might have similar structure, and they might be binding to the similar set of proteins in human body. So the way then to think about this representation is that diseases and drugs are not single nodes in the network. Instead, diseases will be defined as subgraphs of the underlying protein interaction network defined on proteins that when mutated or otherwise altered by genetics or nature, they would give rise to the disease. On the other hand, drugs would be represented as subgraphs of protein interaction network that are defined on drug targets, meaning those are the proteins to which the drug binds in the human body in order to exert a particular function and sort of treat the disease. So in pharmacological sciences, it is known that a drug is likely to have a positive therapeutic effect on the disease if it is close to the disease in some pharmacological space. Okay, so this sounds fantastic, but what does it actually mean? And so our core idea will be that we'll actually use the power of the paradigm of embeddings to operationalize this concept of closeness between diseases and drugs in pharmacological space. And so that will allow us to directly model and incorporate some of the inductive bias that comes from the knowledge of drug biology. So you might say, okay, if I didn't convince you yet, why subgraphs? You might still be asking why subgraphs, and I will show later today in the second part of the talk how some predictions that we made for repurposing of drugs for the novel coronavirus resulted in predictions that were shown to successfully reduce viral infection in wet lab experiments, and 76 out of 77 77 drugs predicted by these methods are such that they don't directly bind to proteins targeted by virus, and instead those drugs that were shown to successfully inhibit viral growth rely exactly on this mechanism and notion that the way drugs work is based on some network effects and network propagation of effects that the drug has, in the sense that the drug binds to its proteins, but then those effects propagate through the underlying biological network. And without a subgraph-based representation, it would not be possible to discover those drugs that successfully reduced viral infection. So it's not just a nice theoretical framework, but it also probably shows to be able to discover new biology, and that would otherwise not be possible to kind of identify with traditional technologies for drug repurposing. From the computational standpoint, why are subgraphs challenging? Learning over subgraphs is challenging because we need to predict over structures of varying size. So these subgraphs, which in our case will be drug and disease subgraphs, can be of different size. They can have different number of nodes and edges. They're not necessarily K-hop neighborhoods. They have rich connectivity patterns, both internally within a subgraph as well as externally, the way they interact with the rest of the biological network. So how can we inject this information into a GNN architecture is the second question. And a third point is that because these subgraphs are given to us as part of how we think of drugs and diseases, it turns out that these drugs can be these subgraphs can be localized, meaning that they would reside in one region of the graph, such as this example on the left part of this picture in the bottom of the slide, or the subgraphs can be distributed across multiple local neighborhoods, just because a drug might work by actually binding to proteins that are very different from each other, which would then mean that the subgraph for the drug would be highly distributed. So the problem to address this challenge, we will formulate the problem as follows. So our goal here will be to learn to specify a transformation function that will learn subgraph embeddings in such a way that the likelihood of preserving subgraph topology will be maximized in the embedding space that we will learn for subgraphs. So intuitively, the way to think of it is that our input is a large biological graph that is overlaid with a large number of subgraphs. Those subgraphs might be complex in the sense that they might actually consist of multiple disconnected components, like S4 consists of 2 disconnected components. And the idea is to take each and every of those subgraphs and be able to embed it into this d-dimensional space in such a way that 2 subgraphs that have similar topology should be embedded close together in the embedding space. Okay, so what does it mean for 2 subgraphs to have similar topology? And so that's what the sub-GNN method really focuses on and tries to kind of study and kind of quantify different aspects of subgraph topology. In particular, in the context of subgraphs, we might want to look at 3 distinct aspects. So we might want to understand what is the neighborhood what is the neighborhood of the graph where a particular subgraph resides. Then we want to know what would be the structure, what is the structure of the subgraph in the sense of whether there are certain higher order structures that are overrepresented, found more often or less often in the subgraph relative to what we would expect otherwise, and where exactly that subgraph can be found within the context of the entire graph. Is it found in more towards the core of the large graph or on the periphery? And so all these 3 properties can be then studied in 2 different ways, whether we're looking at the neighborhood structure or position that is internal to the subgraph, or that really is external to the subgraph, or it is on the border of the subgraph. And so sub-GNN will then is a representation learning framework that will generate embeddings for subgraphs capturing um, these three key properties of subgraph topology, both from the perspective of internal structure as well as external or border structure. Just a very quick overview, what SubGNN does. So it has two key parts. In the first part, the idea is to the idea is to hierarchically propagate neural messages in the graph. And the way that we are doing that is that we are propagating messages from helper subgraphs, which we call anchor patches, to subgraphs that we want to embed, and then we aggregate those messages that each subgraph receives from its anchor patches to get the final subgraph embedding. And this is done through three different channels that route messages according to three different aspects of subgraph topology, as we have seen in the previous slide, in the context of position, neighborhood, and structure. So very, very briefly to unpack these two key parts of SubGNN, the idea is the following. In the first part, we specify neural messages for each of the three aspects of subgraph topology, neighborhood, structure, and position. Those messages are propagated from anchor patches to subgraphs. So in the context of this particular example, imagine we would want to embed this subgraph SUP1. And so this SUP1 subgraph would receive a number of messages that were sent to them from anchor patches. Now, different kinds of anchor patches are defined based on the three aspects of subgraph topology. And so then when those messages arrive at the subgraph SUP1, they're being aggregated in three different ways based on the aspect of subgraph topology, and then this entire process is repeated several times to finally arrive at the final embedding for the subgraph. And so when I said that we have different messages for different kinds to capture different types of network structure, that essentially means that we have three different channels for capturing position of subgraphs relative to the entire graph in which subgraph resides, to capture the neighborhood of the subgraph as well as its structure. And so each of those channels defines a separate set of neural messages and separate it has a different uses a different definition of anchor patches and sampling functions to sample those anchor patches from the graph, as well as to measure the similarity between anchor patches and the subgraph that we want to embed. And so the final result of those three channels gets aggregated, and then this gives us an embedding for the entire subgraph component. If subgraph has multiple components, then the second phase of aggregation kicks in, that then results in the final representation of a subgraph C sub S. Okay, so with this particular methodology, we are able to then embed a large number of subgraphs and learn embeddings from them. So we were thinking of using that for the problem of drug repurposing. So to do that, the first thing one needs to do is to construct a data set. So we collected and processed and prepared a large training data set for the problem of drug repurposing that consists of the underlying protein interaction network that's called from around 50 knowledge bases. It's a reference human interactome, and it has drug on top of that graph. There are many drug protein and disease protein links, and then information about what are treatments of drugs that are currently approved and successful. So this results in exactly this kind of representation and conceptual image that we have seen before, right? So we have this protein interaction network with white where each node indicates a protein. On top of it, we have a large number of disease subgraphs and drug subgraphs. And so the way then to think of this problem is that our input data will actually be a bipartite graph where we don't have nodes, but we really have subgraphs. We have a large number of diseases, a large number of drugs. For each disease and drug, we have its own. We have a separate subgraph, and they are linked based on whether drugs are currently used to treat the disease. So that is the data that is used to train the model, where the model is first taking disease drug subgraphs to learn embeddings for them, and then once the embeddings are obtained, it combines them. It combines those embeddings to predict links actually between drugs and disease subgraphs, where those links are indicative of whether a drug should be indicated for the disease or contraindicated, meaning it was likely to treat or not treat the disease. Using that algorithm on the drug repurposing data set yielded a large number of predictions, and then we pursued various ways to evaluate those predictions, including all standard cross-validation setups. But I would mention here another test that we did with the Spark Translational Research Program at Stanford, and that program has repurposed a relatively large number of drugs over the last 2 decades. And so what they shared with us is they shared with us a list of drugs and diseases where you can think of where you can read this list as a list of successful drug repurposing, repurposing of drugs for the diseases. So let's look at one particular prediction from the bottom. The way that prediction was the model predicted that carvedilol will treat would be useful for treating Chagas disease, and SubGNN model has ranked carvedilol as 9th most highly ranked drug for Chagas disease out of 5,000 drugs that were in the system at that point in time. So you can imagine that. Yes.
So we agreed that we could ask questions during the talk, and there's a question that's coming up now at the Q&A. So the question is the graph used and the relative features publicly available? By Luca Cappelletti.
Yes. So yes, the graph and relevant features are publicly available, and we have actually released them as part of the Therapeutics data commons, as well as another paper that we have out there.
Thank you.
So yeah, we were just looking at returning to the slide. Looking at carvedilol as an example prediction, it was prediction where that were ranked by the model as 9th most promising prediction for Chagas disease, and indeed carvedilol was repurposed for Chagas disease as part of within the Spark Translational Research Program. And even if you look at this example, the other examples shown here in the slide, you can see that predicted ranks are relatively low, meaning that the correct drugs that were repurposed for these diseases were ranked very highly, very close to the top of the list, which indicates good performance of the model. That has led to follow-up research on prostate cancer with the goal of identifying successful drug cocktails that would minimize incontinence, a side effect, and maximize the response of patients, as well as schizophrenia, which is a particularly challenging disease because its disease subgraph is very large, as that disease affects a large number of core biological processes, and so its disease subgraph is quite complicated. It's very complicated to handle. Okay, so let me now move to the second part of the talk, which will still be about identifying drug treatments, but with a different angle in mind. So far, the diseases that I have mentioned and been discussing, and the disease to which the method was applied to, were relatively well-studied diseases. So those they might be very complex diseases for which there is no, they don't yet have good treatment, such as Alzheimer's and Parkinson's, but there's lots of other auxiliary data about those diseases available. There is another way, and another important question here, which is that of what to do about neglected, emerging, or these novel diseases that we have never seen before. And so that will be the motivation for our second part of the talk, and I will also describe a method that allow us to address some of these questions in the context of using meta learning for graphs. So let's start with the motivation. So the motivation for this work.
Oh, so we can't hear you anymore. Can you talk again? So we missed a piece of what you said.
Okay.
Now you're back, I think. Can you say something?
Hello?
Yes. Yeah, I think your microphone is a bit far away now.
Okay.
Something happened to the microphone. Can you speak?
Can you hear me?
Yeah, it's a different sound now. The previous sound was better, but it's okay. We can hear something.
Okay, let's see if this works. I'll go back and share my screen.
Yeah.
Can you hear me well and see the screen?
Yes, we can hear you. Thank you.
Okay, great. Thank you. This part of the work was motivated by the pandemic earlier next year, where we were tasked in early March, actually, with the question of using the AI toolbox that we've developed in an effort to identify repurposing opportunities for COVID-19. And so that, of course, was very important. Repurposing was most promising strategy for identifying and rapidly identifying therapies for the disease, because traditional iterative development and design of a new drug from scratch was simply not feasible. And so the question then becomes, how can we identify therapeutic opportunities for this new disease that we have never seen before, for which very little data is available, and obviously no successful drug treatment. And so that was the motivation for some of the methods that we have developed in this space. And then those methods were successfully then applied in the span of actually 3 months, going from having just input data and predictions to actually experimentally validating those predictions in the wet lab and identifying what drugs that are predicted by the model successfully inhibit virus. So I will explain and return later to this particular application and as well as the results. So the key technical challenge here is that we were tasked with making prediction for this new class, and solving this new task of the disease that we have never seen before, and that there is very little data available. So the challenge that underlies this question is that generalizing to this new class of phenomena, or generalizing to a completely new disease, is very hard. Prevailing GNN methods still required abundant label information. And so prevailing methods would assume that what we have these graphs, and many of its nodes or subgraphs are labeled, meaning we know and have successful examples of drug disease treatments, as we have seen in the first part of the talk. However, that rarely happens in real world, in situations where we really want to use these methods to answer completely new scientific questions. And an example that we are using today of emerging pathogens and emerging diseases is just one of them. There are many other, I would argue, most of the core exciting questions at the frontier of medicine and health are of that kind, that there is very, very little labeled information available, including, for example, if one would be working on novel drugs in development, or one would want to diagnose patients that are hard to diagnose otherwise, in the sense that they have rare diseases or diseases that don't match well with their genomic information, as well as emerging pathogens. And so our first, our question on that route between March to July and then in the summer was to also develop a method that would allow us to really be able to make powerful, accurate predictions for COVID-19, even though essentially no label information for COVID-19 was available that one could essentially learn from. And so the question that underlies that was how to really design powerful meta-learners. And so our key approach, our key idea for that was resulted in an approach that we call GMeta. It stands for meta learning for graphs using local subgraphs. Again, we see this notion of leveraging subgraphs as a way to pull together local interaction information, which in biology is tends to perform really well and is very useful. And so the idea of using subgraphs here for meta learning was motivated by, essentially had two motivations. The first motivation was that of scalability. So traditionally, neural message passing is defined over entire graphs for in order to achieve meta learning, and some of the earlier translations of MAML to graphs aimed to learn signature functions that were signatures of entire graphs, and then use those signatures to translate and transfer information from one graph to the other. So our observation was that that is not particularly useful because first, it dilutes much of the signal, and second, when we have a large number of graphs, which we had thousands of large graphs from different species, then it also does not scale well. So our idea was, let's instead use subgraphs and learn subgraph level signatures, and then translate those subgraph level signatures from one particular test to the other, or one graph to the other. And so what is the value of those subgraphs? So the way to think of graph structure versus node labels in the context when little labeled information is available is the following. So intuitively, we can think of, there are two sources of information that GNNs try to leverage. One is this notion of label propagation, where the assumption is that nodes that have the same label or similar properties, or diseases that have similar phenotypes, or drugs with similar treatments would tend to be nearby in the graph. And so if that's the case, then label propagation might be an effective strategy. However, when there are not many labels, we cannot leverage label propagation so well. So in that case, we might want to rely on structural equivalence or structural learning, where we would leverage more strongly structure of the graph, in the sense that nodes that might, or that have the same label, might not be necessarily close to each other in the graph or proximal to each other in the graph, but they might have similar network shapes. So similar number of triangles, squares, other kinds of motifs or graph structure that would make those nodes similar. So the idea was, when labels are scarce, such as in the case of COVID-19 repurposing, label propagation is not particularly useful or meaningful. So can we leverage structural equivalence and graph structure better? And so that's exactly what GMeta can do. And I will not go into details here because of the lack of time, but the idea is that under certain assumptions, we can show that these local subgraphs that we define around nodes and that we use to represent drugs and diseases, they capture most of the useful information for predicting, for making predictions of properties of those subgraphs. And they preserve near the same feature information as if we would do label propagation across the entire graph and then characterize and be learning graph levels signature functions. So now we don't need to do that which makes the model much more makes the model more scalable, as well as it makes it turns out it also performs better. So going back to our application of how GMeta can then be used and leveraged for the problem of drug repurposing, let's focus on that for the next few minutes. So our task was, or sort of our work started the beginning of March last year. At that point in time, we had a human interaction network and information on what are the proteins that the virus targets. And so between March and April, we used graph neural networks and some of the approaches that I mentioned just before to identify a ranked list of drugs that were predicted to be promising therapeutic opportunities for COVID-19. Those were then experimentally screened, and here we will see where the images actually come into play between April and June. And then in June, then some of experimental readouts were generated and further validations were done. So starting in March, the first question was how to represent COVID-19. What like computationally, what could that be in the context of graphs? So the way to think of it is the following. So we have already seen today human interactome, human protein interaction network, which is this network where nodes are human proteins and edges indicate whether two proteins bind in the body. So when the virus comes, virus and its proteins, virus has its own proteins, those proteins would bind, would target some of the proteins in human body. And so those proteins that are targeted by the virus, those human proteins that are targeted by viral proteins, can be used then to identify viral disease module or COVID module, shown here in this gray color. And so what we will do then, we will actually think of COVID-19 just another subgraph, another module in our graph, in addition to having a large number of drug modules. Okay, so if we actually do that, this is how COVID-19 disease module or COVID-19 disease subgraph looks like. It's a subgraph of 332 human proteins that were identified experimentally through the work that Gordon et al. At UCSF did, where they have actually expressed 26 of 29 SARS-CoV-2 proteins to identify which human proteins those viral proteins attack. And so that resulted then in this COVID-19 disease module. So using this particular COVID-19 subgraph, we were then, in addition to subgraphs for all approved drugs in the US, we then use that to train essentially a modified sub-GNN model with the meta-learning twist in order to be able to produce a ranked list of drugs that were predicted to be good, promising repurposing opportunities. And so using those predictions, the first question that we wanted to ask is just computationally, can we get sense how good those predictions are before we pursue experimental validation in the lab? And so for that, we were monitoring drugs under clinical trial during that period of time. And those drugs that were under clinical trial are, according to medical community, potential good, useful drugs for the disease. They are of course not yet approved, but they are a good proxy of what a ground truth information might look like. And so we use that proxy information to be able to test and measure the accuracy quality of predictions generated by graph neural networks and disease and drug subgraphs, and compare that with a number of classic drug repurposing methods based on network science, random walks, diffusion, and so on. So that led to substantial improvement as measured by AU-ROC in terms of GNNs being able to more correctly pinpoint what at that point in time were considered most promising drugs for COVID-19 and were studied under clinical trial. But really, that is good computational strategy to make sense of predictions. But down the line, the main question becomes, well, how can those predictions really inform downstream experimentation in the lab? And so to do that, we have we've been part of a large task force here in Boston and at Harvard on COVID-19, and our collaborators from the National Infectious Disease Laboratories were actually designing a screen to make sense and test some of the predictions, and especially those predictions that were predicted by the model as top-ranked predictions. So in order to do that, the idea is the following. So the idea is to take predicted drugs and put those drugs in a petri dish where there are cells infected with the virus, and then wait for some time and see whether those cell cultures where drugs with drugs that were predicted to be most effective really tend to survive for a longer period of time, and really the infection is halted, which would indicate that by introducing these new drugs, introducing drugs to the cell culture is useful for halting or reducing infection. And so the idea here is to take a culture of cells, first use an antibody that detects whether in each cell the virus is present, yes or no. And on top of that antibody, add another antibody that would fluoresce under blue light. And so then essentially one can take photos of those cell cultures and analyze the amount of fluorescence as well as location of that fluorescence color in the images to get sense into whether the infection was reduced or halted. So the way this looks like, it looked like the following. So
Marinka, quickly, so you have now talked 45 minutes. So just for
yes.
so that's about the time, but
thank you, Max. I'll have just a few more slides and then I'll wrap up. And so the design here was the following: to take these large wells of large plates of wells where drugs or compounds were administered to cells and to cells that were infected to the virus. And then those wells were incubated for 48 hours. And then images were taken of the wells. And for those images, we analyzed them to identify nuclei, to identify cell boundaries, and actually measure the presence and the amount of virus after 48 hours. So that information and extracted features can then be used to define dose response curves, which are fitted against the features extracted from the images in the effort to reliably estimate what is the amount of viral infection that was reduced. And so that is then the readout that indicates how successful a predicted drug would be based on how much of the viral infection has is gone after 48 hours. So our collaborators were using that approach then to take 900 compounds that were ranked at the top of our predicted list of drugs. And so for those compounds, they found 77 with strong effect on the virus across a broad range of concentration, and that was an order of magnitude better hit rate than what would be possible by using a simple brute force approach, which would be just screening all drugs in the lab for their response to the virus. Instead, focusing on top 100 drugs that were ranked that were found in top 100 list of our predicted drugs led to an order of magnitude a better hit rate than what was possible before. And so these sets of novel drugs that we refer to as network drugs, 76 of those 77 identified drugs are such that they don't really directly bind to proteins targeted by SARS-CoV, but instead they bind to and target some other human proteins that indirectly interact with proteins that are attacked by the virus. So they really operate on network-based actions that could not be otherwise learned. I'll wrap with key ML lessons here. So what we learned during this process of very close collaboration with domain experts is that it's extremely important for domain scientists to be able to interact with AI systems and somehow provide feedback to the ML loop. It was important for us where essentially progress had to be made on a daily basis in terms of new predictions, novel characterizations, to inform downstream experiments in the lab. The domain experts were able to get sense out of predictions, ask questions that would yield explanations in those predictions. So we worked with some of the earlier methods that we developed based on GNN Explainer and others that can provide explanations together with predictions, which turned to be very useful for domain expert to build trust into predictions. Still, the key lesson is generalizing to new diseases. To a new disease is generally very, very hard, much more challenging than making predictions for a disease for which there are already some treatments. Okay, so I will conclude here. I mentioned that we have all different data sets and learning tasks for therapeutics available. And so that's part of our effort that we call Therapeutics Data Commons. If you go to this website, you will see currently a collection of 66 data sets spread across 22 meaningful machine learning tasks in the area of developing therapeutics. Imaging data will come soon as well. And so these data are ML ready. They come with meaningful data splits as well as leaderboards in an effort to facilitate algorithmic as well as scientific innovation in the area of therapeutics. I'll stop here and I'm happy to take any questions.
Thank you very much, Marinka, for a wonderful talk. It's an amazing complexity that you displayed here. To get everything working on COVID in such a short time is like very, very impressive. Are there questions from the panelists that you, let's see, you can put them in the Q&A. There's one question here by Case Elbers, which I will read for you. So hi, I have a question about sub-GNN. Do you see a way of accounting for causal mechanisms in graph embeddings or inferring them? Could you treat directed signaling protein interactions, such as protein phosphorylation, differently than proteins that bind to each other to form a complex?
So that's a wonderful question. Sub-GNN currently cannot account for causal mechanisms. That it would be great if that would be possible to do. One of our ongoing efforts is actually goes in that direction with the goal of identifying drug mechanisms of action that would that make one step towards identifying causal mechanisms of drug treatments. But that's currently something that SubGNN does not support. Regarding the second question, whether we could use some other kind of biological networks instead of protein-protein interaction networks with SubGNN, the answer to that is, of course. In the experiments and much of the analysis that I described today, the underlying network was always human protein interaction network. So we would replace that network with a signaling network that would indicate protein phosphorylation as suggested by Keith. Our expectation is that those networks, since they are typically understood as being more causally indicative in biology, would be able to really identify better potentially causal, or would correlate more strongly with causal mechanisms than what is currently possible with PPI networks. But that is kind of, there's open questions both in terms of the data sets, that one should use here, as well as the methods that we don't yet have that would be able to learn causal embeddings or in some way be able to work with that. The best we have is we're able to generate explanations that use counterfactual inference to identify small subgraphs that when removed from the data, they change prediction by a large amount, but that's still very far from kind of causal reasoning that we want to get here eventually.
There are some methods that combine sort of interventions and sort of both, so they combine interventions and basically observational data into one mechanism, and then you can sort of learn the causal direction better, I guess, if you include that. So here's another question from Gonzalo Valadão. For a rare disease with known biochemistry, but no treatment yet, what approach, the first or the second, should be more suitable? Thank you.
So the second approach that I mentioned today would be more suitable, that of meta learning and transfer learning to a new disease, because for the first approach and SubGNN that I was mentioning, it primarily relies on the assumption that we already have some examples of successful treatments, and those can be leveraged to then make predictions about additional treatments that might be suitable for treating a particular disease. So when one would have a rare disease that is studied well, but no treatment yet exists for that, and I think yesterday or the day ago, 2 days ago, there was the Rare Disease Day, which is very important day to recognize this prevalence of rare diseases. Then one would want to use transfer learning, meta learning strategies, because lots of data is available. That data biochemistry data might be used for transfer learning, but no labels are there. So biochemistry data can be used to kind of almost estimate similarity between this rare disease as well as other diseases that already have some treatments, and then transfer that information effectively. So GMeta would be a better approach for such a disease.
Thank you. I actually had a question myself as well. So you sort of assume that you have the subgraphs. Or so is there a way that you could actually learn to generate the subgraphs dynamically or from the data?
So that's a great question, and I've received that question several times already. Yes, our assumption in all of the applications I mentioned today was that subgraphs were given to us. Those are descriptors of drugs and diseases. We describe them through these rich subgraphs. And that is okay for drug repurposing, but this question of generating subgraphs is a very, very valid and exciting one, because our current knowledge of drugs and diseases is incomplete, meaning the subgraphs that we currently have are incomplete. And so it would actually be of great interest to be able to take existing subgraphs as kind of seed representation and expand them, or which in the form of machine learning that would translate into identifying novel drug targets, if you would be expanding drug subgraphs, or identifying novel disease genes or disease gene association discovery, if you would be expanding disease subgraphs. So yeah, it's a great question. We haven't tackled it in the context of drug repurposing. We were primarily focused on recovering relations between drugs and diseases, but separately, independently of that, drugs and diseases and their knowledge is incomplete and could be expanded. And the way to do that would be essentially to think of either completing, growing, expanding those subgraphs, or generating them from scratch. Yeah.
I have one final question: whether you envision in the future you can actually close the loop completely and have basically robots do the experiments for you, and then no humans are needed anymore, right? It just everything goes completely automatically, including the experiments, and just the right drugs come rolling off the
yeah.
the
So
band.
that's a great vision to have. We are making some strides toward it, because based on what we learned with this very quick COVID work, is that the largest gap that we now see between kind of core ML methods and the use of these methods for novel discovery is in this part of communication or translation of predictions to an interaction with domain experts. I would think of that as this part of generating actionable hypothesis, which I had on second slide, where actionable means that the prediction is such that directly translates to testable hypothesis. And so let me give just an example. If you work with biologists or clinicians and say, well, I have this amazing camel method, wonderful, it's great AUROC, it outperforms everything else out there. And it makes, the model makes the prediction that drug A will treat disease B. So that's still a very, very high level prediction. So how exactly to go about it? So we are thinking about now having ways to answer more complex logical queries on these graphs, so that the model would produce an output that would not be just a drug will treat the disease, and like how will that do? Nobody, or very few people will take that prediction and now commit millions of dollars and lots of money and resources to validate such a high level non-actionable prediction. So the idea here is to move towards being able to say that drug A will predict disease B, and the prediction is that it will do that by binding to these proteins in human body that in turn interact with two other proteins that in turn are associated with disease, right? And producing that as output, it's more actionable prediction that can then really more directly yield to these downstream experiments, which in many ways are already today done through automation and robots who move around these plates that I was mentioning in the second part of the talk, and kind of take images of the cell cultures and analyze them.
All right, thank you so much. It's 5 o'clock here. So thank you again for an extremely interesting and exciting talk. So I would like to also thank all the speakers of today's session, but I don't want to forget one person who has organized this, which is Dan Lee. So I'm not sure if Dan Lee can show herself, or maybe she doesn't want to show herself, but she was absolutely instrumental. There she is. So Dan Lee, thank you very much. Setting everything up and getting the Zoom link and everything to work, and the website and everything. So thanks to her, we had this meeting today. And that ends today's workshop. And I wish you a great rest of the day, and hope to see you next time again. Bye-bye.
Bye.